Mike is he's a two-time alumnus of EKU. He graduated with his bachelor's in biology, or actually, I guess, fisheries management at the time with Franklin Grandson when he was here. And then, as Brady said, he worked with Dr. Schuster on his master's degree after that. He's been employed with a couple of different Kentucky agencies. Currently, as you can see, he's with Kentucky State Nature Preserves. He's been with them for 11 years. He's his area is aquatic conservation. He deals with mussels and snails, microinvertebrates, fish, and oversees a lot of the projects relative to conservation. He's also worked with Texas Tech University and with Daniel Boone National Forest. He's also been with Kentucky Division of Water, and when he was at Division of Water, he was one of the main ones that produced they had a bioassessment of fishes, and it's the Kentucky IBI, and so he was an integral part of that. He also worked with a lot of other species, imperiled species, a lot of fish species like the red-lipped darter, which was part of his some other work that he was doing from when he was at Texas Tech. And then he's worked with black-side dace, arrow darters, and the relic darter. And just recently, with U.S. Fish and Wildlife Service, they were able to downgrade the relic darter down to from endangered to the threatened. Couple of current projects: status assessment, conservation genetics of the Kentucky creek shell, and monitoring of the Green River associated with dam removals. And that's been a big one lately. I know I've heard that there's a third dam coming out this summer, right? Yep. And so Mike's gonna today focus on the mussel component of the Green River dam removal project for his seminar.
Sure. Really happy to be here. As they mentioned, I'm an alumni, and it's exciting to present in front of a lot of familiar faces and a lot of new young faces too. So today I'm going to be talking about some dam removals that are going on or pending dam removals on the Green River. This picture shows the location of the former location of Lock and Dam number 6 that was removed in 2017, and Lock and Dam number 5 is pending. That should get removed hopefully later this summer, actually. Before, where is it? Switching. Got it. But there we go. Before I start and get deep into this, I want to thank a bunch of people. As I mentioned to Sherry a little bit ago, conservation is kind of like a team sport. You just no one person can do it. It really takes a group effort, a lot of people working together on a similar type goal. And there are numerous people I left off here, but these people are very, very helpful in so many ways. So I just want to make sure I say thank you to them. So let's talk about dams and the effects of dams. To my surprise, as I was doing some research looking into this, there's nearly 100,000 dams. There's slightly over 90,000 dams in the United States. Most of them are small dams. Kentucky has 1,100 dams. That surprised me a whole bunch. Most of the times when we think of dams, we think of like Lake Cumberland, the LBL area, where there's Lake Barkley, Kentucky Lake, where there's really large, massive dams, or the series of dams on the Ohio River. But the overwhelming majority of dams in the state are small dams. Kentucky has hundreds of streams called Mill Creek. And a lot of that is associated with a mill that was put in there, whether it was a sawmill or a grain mill, where they put a dam in there to run that and proceed. So many streams in Kentucky have had dams of various size in them, and they've had a lot of impacts, and I'll get into that in a minute. But dams also have had a lot of benefits for us in society, and as we progress and move forward over the centuries, really. And some of the obvious benefits are for navigational purposes. The Ohio River has a series of lock and dams that was set up to help the navigation up and down the Ohio River. The Ohio River, without the dams, is actually somewhat of a shallow river. There's areas in there where there's a lot of shoals, pretty shallow, a boat couldn't navigate that. So they got the novel idea to put in some dams. Unfortunately, I'm not supporting of dams, so I don't want to mislead anyone, but that was the rationale behind it. Flood control, people like to live on the floodplain, so having the dams helps control the flood situation during rain events. We get hydropower. There's recreational activities such as Lake Cumberland, as well as water supply. Numerous communities along the Kentucky River, they get their water from the main Kentucky River, so having a dam allows a nice pool to be the source for the water supply. However, with these dams, there's a lot of impacts that they can cause on a aquatic ecosystem. Before I dive into that, they're also a safety hazard. Dozens and dozens of people die every year associated with dams, not necessarily the real large dams, but the smaller picture, the lower picture of the smaller dam is an example of a lot of an unsafe dam. A lot of people get trapped in the undertow and they end up drowning. So they are a safety hazard, and it's something not to take lightly. But ecologically, they drastically alter the hydrology of a stream. Streams are loaded systems, flowing water, and when you put a barrier such as a dam up, that takes that flowing water, makes it standing water. That changes things dramatically. Obviously, the habitat goes from flowing to standing, but also the water quality changes, the temperature, pH can change in those situations, as well as the sediments and substrates that get transported into the system. They no longer are transported. Streams streams have three main functions. They erode, they transport, and they deposit material. And so when you have a dam up there, that interrupts those three functions. Dams also are a major barrier, especially for fish, to where they can't disperse or migrate properly. And they also affect, due to the change of the hydrology, the drift component with invertebrates, where invertebrates are drifting the water, they're a food source. Also, larval fish will drift as they mature, and having a dam will impede that. And then the most obvious one is decreased biodiversity. So we have primarily mussels and fish. It's well documented where the diversity of those organisms decrease with the presence of dams. So on the Green River, the dams we're focusing on today are Lock and Dam number 5 and number 6. They were built around the turn of the century. Lock and Dam 5 was 1899, and Lock and Dam number 6 was completed in 1906. The picture that you see here, that's of Lock and Dam 6. It's a smaller dam. It was only about 10, 15 feet tall. It was put in there for navigational purposes by the Army Corps of Engineers. And in the 50s, Lock and Dam 6 was decommissioned. And over the decades, it fell into disrepair and became kind of a sieve a little bit. It still was a barrier, but it was no longer the perfect dam anymore. So its removal was pending for decades. And actually in the 90s, Dr. Schuster had a student named Greg Pond, and his master's project was to look at the macroinvertebrate community within Mammoth Cave National Park associated with Lock and Dam 6 and its pending removal. So it was a baseline data set for his thesis. So that was some of the early work that was done on the Green River associated with these dam removals. But in November of 2016, a weather event occurred and Lock and Dam 6 breach caused a major safety hazard, and that spurred multiple agencies getting together, primarily the Army Corps of Engineers, U.S. Fish and Wildlife Service, to remove Lock and Dam 6 officially. And along those lines, that then spurred conversations of other dams in the Green River to be removed. And so the hydrology in the Green River with these dams. So this yellow dot is Lock and Dam 5, the green dot is Lock and Dam 6. The red line shows the impounded waters due to the dams. The yellow segments show the transitional. During high water, the river is impounded. During summertime, low water, it's a free flowing. So we call that the transitional zone of the river. And then further upstream in the green, you have free flowing river conditions. And then below Lock and Dam 5, you have free flowing river conditions, but during high water, you get the impact a little bit of inundation due to Lock and Dam number 4. So when 6 was removed, it freed up this segment. This was transitional. That became all now free flowing, and a little bit down this section became free flowing. That little bend there where it turns green to red is an island that we call that Crump Island. Now I'll mention Crump Island numerous times as I go forward. And the red shows 40 kilometers of impounded river. So Lock and Dam 5 is such a large dam, it impounds the water even beyond the influence of Lock and Dam 6, or beyond the footprint of Lock and Dam 6. And so it's a pretty massive dam, as you can see here. So last year they began the deconstruction of Lock and Dam 5. Due to weather and other circumstances, they weren't able to complete that. They're going to start that up come June, pending weather. And hopefully in 6 to 8 weeks, it will be removed then, once they commence. But Lock and Dam 5 is a much larger dam. It is roughly 30 feet tall. So what's the significance of removing these? A lot of it has to do with the actual location there in the Green River. Mammoth Cave National Park is the upper part of the impounded area is within the Mammoth Cave National Park. So it's a important matter for them to see these removed. Potentially can help benefit the springs and the cave systems within the park, let alone obviously the main stem of the Green River. And the main stem of Green River is one of the most biologically diverse systems that we have really in the temperate zone. The only other systems that might be more diverse, drive a little bit farther south into Tennessee and Alabama, and you can get greater diversity than what we have here. But this is really high diversity, and it's phenomenal, really. And so having it impounded was detrimental to these groups of species. So the removal of the dam has the potential to provide a lot of recovery for species. There's over 15 federally listed or petitioned species within this section of the Green River. So what benefits will we see from this removal? And we should see increased diversity amongst multiple groups. We also should have the assemblage be more indicative now of a free-flowing system rather than a lake or a pond type situation. The fish will be able to move freely. There should be better gene flow going on. We should also see improved water quality, cooler temperatures, and overall better habitat and greater diversity of habitat will benefit. So these should be, based on past studies and things, these should be the things we see. But it was asked to me, well, how do we know? We can't just remove the dam and say everything's be hunky-dory and walk away, especially when there's multiple million dollars going into these dam removals. And so the goal is to hopefully show these benefits and measure those types of benefits and then be able to really help support future dam removals too. Not only justify the removals of Lock Dam 5 and 6. So this is a huge project. And so of the goals, ultimately, we just want to document what are the pre-dam removal conditions. Now, what is this baseline data set going to be? Because down the road, 5-10 years from now, we're going to need to compare that time frame to something prior to the dam being removed. So this is what the gist of the work that I'm doing right now. So trying to get this baseline data set, looking at these multiple groups at the communities, as well as looking at habitat. And I've just added, I'm trying to start a gene flow study looking at some select fishes and select mussels, to where the fish host mussel relationship. I'll pair those species up and look at the genes prior to the Lock Dam 5 being removed, below the dam as well as above. And I'm trying to work out the collaboration there with a professor down at Austin Peay State University. Also just started this past February a little water quality study. We haven't done any water quality work, and I was able to get some Kentucky Division of Water people as well as Army Corps of Engineers folks to partner up and tackle a water quality project. And we're going to only do 2 sites. I'm going to use CREP. CREP is a program that, I forget the agency that started it, maybe the EPA. And they gave money to farmers to help manage the land better. So there's a historical sites on the Green River, which we will use for the pre-dam removal data. And then for the next few years, we're going to get current data. So in the long-term study aspect, here's the study area. And you see Lock Dam 5 and 6. I've broken the river down into what I call treatment segments, different segments of river based on the presence or absence of the dam and whether you're above or below the dam. And so you had 5 impacted segments and 1 control segment. And the control segment to the far right there is not influenced by the dam in any aspect. It's completely independent from the dams, where the others have some type of influence at some point in time from the dams. So I'm going to get a data set of biotic and abiotic data prior to the dam, and then ultimately make comparisons down the road post-dam removal. So talk about these segments. Eventually I'll get into these mussels. So sorry if I'm going a little long here. So the first segment, I'm going to start downstream, work way upstream, is what I call Green River below Lock Dam 5. And you see right there, Lock Dam 5. This is roughly a 6-mile long segment. It's kind of a deep run habitat, and there's not really well-developed shoals. Shoals are the most shallow, fastest flowing water. This tends to be pretty swift water, but also deeper. And that's kind of its characteristics. The banks do tend to be fairly stable with a lot of trees and rocks on them. The next segment, and this is the one immediately above Lock Dam 5, above that 30-foot dam. This is the longest segment of stream, and it's 13.5 miles. It is also the deepest. It's over 30 feet in a couple spots. However, there is some flow going still through this section of the channel. So there is a small thalweg where we can find some coarse material rather than just sand and mud. And there's also some rocky bluffs that form nice ledges, which might be beneficial for some species of fish as well as mussels. Next one, and we start to enter into Mammoth Cave National Park. This is now above Lock Dam 6. And this segment goes all the way upstream to Crump Island. And it's almost 7 miles in length. It's a little bit shallower as we go farther upstream, but it's still impounded. There's also, very noticeable with this, a lot more woody debris is found in the river here. And then we get above Crump Island. And so this is part of the river that was inundated back in 2016 and prior. But when the dam breached, the water drained out very quickly. And so you have it much shallower now. The segment is roughly 6 miles in length. This is now free flowing, but there's a lot of sand, a lot of soft material, but there is a good amount of coarse substrates. However, some of those sections are unstable. And my control segment, no, I'll get to the Nolan in a second, but my control segment, which is on the green, is unimpacted by the dams. You have very good shoal development and a lot of meandering of the water, creating a lot of nooks and crannies for things to live in. And the banks are the most stable here, typically a lot of rock as well as good forests around them. And then we have the Nolan River, and this is a tributary from the north that goes into the green. It's much narrower, not as wide as the main stem green. And my reach on it is much shorter than the others too. But it is downstream of the Nolan River Reservoir, and they discharge. And so that scours everything out. So you end up with a very compacted type channel, kind of a V-shaped channel. There's only about a 2-foot band of coarse rock within that channel. Otherwise, it's mostly mud or hard pan clay and a little bit of sand. And there's a lot of trees in there. And that's because these banks, when the water subsided, those banks all collapsed, and I call it bank failure. And they just drop into the river. And this is an example of some of the banks to where when we had high water, this bank was up here, that water got drained, and the whole bank just slumped down. And nearly 100% of the Nolan River exhibited this type of result after the dam got removed immediately afterwards. So there's a lot of sediment then being introduced. And then the other picture is actually from main stem Green River. In the lower section, there's a lot of bank slumping, but not to the extent that we found in the Nolan. So let's talk mussels. That's what I'm here for. So, but that's the quickest route I can go for intro to get you guys up to speed. So with the mussel surveys, and a lot of the other groups too, I took a stratified random approach where I have my different segments, and then I'm sampling random points within those segments. And then I'm not going to be speaking about a given lat long point that I go to. I'm going to be using all of those dots to represent the representation for that segment, that length of stream there, and talk about it at that scale. So we ended up, I zoomed in here. So this photo is, or this figure is zoomed in of a couple segments of river, where this is Lock and Dam 6. This segment is the zone of above Lock and Dam 6 up to Crump Island. And then this is Crump Island there to where it's now free flowing. I had a lot more dots because I broke the habitat into pool habitat and shoal habitat. And then I also have habitat impounded. And so we sampled within these areas. We did 3 transects bank to bank, and all 3 transects were compiled to represent the mussel assemblage for that location. That thing's identified, measured, and we release the mussels back into the stream. So with analyses, we're looking at the overall structure of those assemblages, and I end up using non-metric multidimensional scaling. Followed the McCoon and Grace general procedures used for it, used the Gray-Curtis distance measure. Ended up getting a two-dimensional solution for it. And the final stress was a little high, but 6.7, but still what I would deem as acceptable. I did omit 10 sites where we didn't get any mussels at all. I just left them out of the data set for this analysis purpose. Eventually, I will run multi-response permutation, MRPP, to test differences among the segments as well as among time periods. But since I don't have the time frame yet, I'm holding off on doing that. Also going to look at things univariately, such as richness and abundance with the mussel data. And so from all the work, we ended up doing 88 surveys, got 37 species, slightly over 5,400 individuals. 6 of those species, though, were federally listed or petitioned to be listed. And one of my past assistants, these pictures kind of show the dive setup to where we have a hookah rig or a direct surface air feed. We dive and we're going looking for mussels. This is from a different day, not part of the study, but this is a great example of the setup. We're going look for mussels, dive down, and we're about maybe 8 feet down, and we find Cumberlandia monodonta, the spectacle case, which is a federally endangered mussel. And that was amazing. It's one of those mussels you don't think you ever seen in your life, and then next thing you know, you find it. It's pretty exciting on those types of days. But the dive team that did this, they were going bank to bank, and whatever they got, they added it to that site. So all the species, here's the list of 37 species. I highlighted in orange the federally listed species that were encountered, and you can see their abundance and the overall rail abundance, how they fit within the entire suite of mussels that we encountered. Overall, when I look at the top 5 or 6 species, the overwhelming majority of them, except for Actinidius lignatina, except for that species, the overwhelming majority are really pool tolerant, slack water tolerant species. And they make up close to 45–50% of the community. However, that's for everything as a whole. So if we break it down then by the given segments, and I put in the historical context for what is known within that study area that we have, what are the known mussels. So 61 species have been documented. Of the 61, there's about 10 of them that are kind of on the edge, and there's only 1 or 2 records of them. So I would say within our study area, there's probably close to like 50 species I would expect to find if the stream was like in good condition. But our study, we ended up finding 37. So that's definitely less than even my lowered expectations of roughly 50. And again, that's for everything. But the breakdown then per segment, as you look across, each segment is relatively, this field relatively close to the 37 for the entire system. So some of those sites had nearly, 60–70% of all the species present. Now there were, as you see the Noble River, only 2 species were found. And I'll get into some of this rationale in a minute, as well as above Lock and Dam 6, where that was much lower. So when we look at all these sites, what was the mean richness at each of these surveys that were done? So as we look at the graph here, think downstream, we're working way upstream. So the first 2 bars represent the segment of river below Lock and Dam 5, and then I have it broken down into those habitat categories to where shoal and pool. And you see below Lock and Dam 5, where it's free flowing, much higher diversity. And then as soon as you get above Lock and Dam 5 into these inundated, pooled sections of river, the diversity really drops off. Now there's the bars represent the margin of error, the standard deviation. And then the control site in the shoal habitat is equal for the surveys as when we got below 5. So so the next thing I wanted to look at then was okay, how does this fit in ordination space? How do these sites, all these sample events, how similar are they to each other? Where do they fit? Do we have clustering, or is it completely random scatter shot? And so, I'll guide you here a little, because this is messy, and I have, I'll break it down here shortly. So the pool habitat is represented by the circles, the triangles are the shoal habitat, and the impounded are the squares. And the color represents the segment for things. So when I look at this, it looks really messy. And I decided to break it down a little bit easier. So I only pulled out, I showed the depiction, the exact same data, but I dropped out all the pool and impounded sites and just showing the shoal habitat. And it really starts showing now, we have differences going on with the assemblage structure between below Lock and Dam 5, as well as the stuff that was formerly all green, used to be inundated a couple of years ago. And that is still showing in my mind that it's maintaining that pool community. There hasn't been a transition yet. It's too soon for that community to transition into a free-flowing type community. That will take, it's going to take multiple years, many years. And then our control segment is also unimpacted by any dams, and it is different than the other two suites here. So that's for the shoal habitat. And of the species, when we look at that, I'll start here with the control. When I look at these list of species, this really tells me this is kind of a classic large river or mid-sized river, shallow water, fast-moving water type of assemblage. And these are just the first 5 most common species that I pulled. I didn't pull up the whole list of 20-some per site. But then when we look at below Lock and Dam 5 as well as above Crump Island, you start getting more pool tolerant species such as Megaloneus nervosa, quadrula, Tamarus elatus. Those become much more abundant, much more associated with pool tolerance. Whereas those species are not even in the top list. They're present at the control site, but they're a much smaller percentage of the community. Then when we look at this with the pool and the impounded stuff, impounded segments, it looks awfully messy again. And we'll break it down by the species. I did not add in the control just because there was only 2 sites sampled for it. But the overall message here, when we look at the species, Tamarus elatus, Megaloneus nervosa, quadrula, the same players over and over, and they're pool tolerant. But what is different and why we have a little separation between below Lock and Dam 5 and all the stuff above Lock and Dam 5, if you notice the abundances are very very different, much more abundant below Lock and Dam 5. And this kind of shows the path. So yellow is below Lock and Dam 5, all the green is above, and it's scattered. So what I'm noticing, our conclusion here with this is the diversity, the richness is relatively the same, but the overall structure is really scattered per site within this impounded section, as well as the abundances of whatever species might be there is really low. Whereas the abundances and the overall structure below Lock and Dam 5 is much much similar regardless of site. And in this graph here highlights the differences in the abundances. Again, these are below Lock and Dam 5, much more abundant. Then we get above Lock and Dam 5, all this pool type habitat, those abundances really drop out. And then on the right-hand side, you have the control. And that's pretty abundant. But again, with the control, you have a different suite of species where it's more free-flowing associated rather than these pool tolerant species. So it goes back to when we look at this, even though we have all more or less relative equal diversity, the players that make up that diversity are different among the overall large habitat types. And when we break this down, only looking at the federally listed species, the federally listed P&E species, historically we had 17 known species within the green and within that area. Our study found 6 of them, and below Lucky Man 5, all 6 were found. But as you get to the pool sections, you see 0, 0, 3, and 0 again, to where they're just not present. Or if they are, the one with the 3, that was only 5 individuals. Where below Lucky Man 5, we had roughly 100 individuals, and the control segment had 4 species and slightly greater than 80 individuals total. So when I look at this, I see a lot of potential. Because if the dam gets removed, comes free flowing, in due time, it will get more like the control segment. So this gap between 6 and 17, I view that as the potential for this river to recover. And to where we look at this, wow, we're looking at roughly 10 species we could recover, and then adding that up to the 40 kilometers of river that would now be available for free-flowing habitat. There's a lot of space, a lot of, I think a lot of room for recovery for the mussels within this segment of river. And I didn't say this initially as I got involved in this project, but thinking about it, this really is one of the largest stream restoration projects ever that I know of in Kentucky. And with the removal of 5, Sherry mentioned the 3rd dam is going to get removed, Bear River #1. With this type of activity, there's a lot of hope and optimism to really recover some species that are really, really rare. And either through natural recruitment or through partnerships with the Kentucky Fish and Wildlife and reintroductions of species back into these areas, hopefully in due time, we can start seeing these species again that are just barely hanging on. So some of the thoughts, and kind of, I would hit on this a little bit. Overall, the impounded sections, they have the diversity, but it's the abundance that really is really making things look different versus those free-flowing sections. So all the players are there, there's just low numbers. The impact from the Nolan River, I didn't talk about this too much, but that discharge from the Nolan River really scours out. I don't think there's any real chance until they change the discharge pattern on the Nolan River reservoir for anything to truly get established there, except for a few species. Notable species that were absent is Epioblasma triquetra, the snuffbox, round hickory nut, clubshell. These are all federally endangered, and they are known historically within the green. For some reason, they are not showing up in our surveys. And take any questions this time.
Thank you very much, Mike. Any questions for folks in first? Any questions for those in the second?
Mike,
Yeah.
you guys are sampling each year from here on out, or how often are you going to be sampling?
So I'm working on the proposal. For fish, I plan to get in there immediately and then sample that on an annual basis. With mussels, we'll probably wait a couple of years before going in there due to their life strategies. They're just not gonna, we're not gonna go find a 20 millimeter mussel the next year or anything like that. So that's why in above Crump Island, that segment, even though it was after the dam was removed, as we did the surveys, we're measuring things, those were all mussels that were born prior to that dam being removed. So I have no problem including that as pre-dam removal data in that regard. Anyone else?
I have a question actually. So you just mentioned, I know essentially nothing about shellfish, but you just mentioned some of these were around before dam removal. So what's the typical lifespan of one of these mussels?
So that one mussel showing my sister Evelyn holding in her hand, that's the spectacle case. They can live to be almost 100 years old. Others though are they can be maybe 5 years as kind of a max life for some of them. But in general, search this river of this size, 20, 30 years is very typical for a majority of the species.
And that's, there's a big range across the species there. Is that right?
Yeah,
Okay.
yeah.
Okay.
So Mike, do you have a sense of how long it might take? So check down out, say, in year one.
Yeah.
How long do you think it might take for that part of the river to go back to a semi-natural state?
What I am seeing, I think within a few years, as far as habitat and the flow goes, because what I'm seeing above Crump Island, that the hydrology, the river, it's already trying to do its thing. The physics are already working on it. There's a lot of material that needs to get moved, so there's large areas that are unstable. But I think the biggest hurdles are going to be when the water goes down, these banks are going to slump in. So you're going to end up with that type of sediment load, you're going to end up with a lot of trees in there, which that's okay in my mind, that's habitat. But eventually, all that will work itself out. And I think really, from what I've seen over the last few years, the river is changing every year. It's like a living beast, really. And I think it will start to stabilize. I feel confident to say within 5 years.
Really?
And I think the fish will move in really within a year. I think we'll start seeing a lot of diversity with fish, but I think the numbers will be low and not fully established yet. But in a few years, I expect those populations to kind of get stabilized and become much more abundant and eventually see the shift in the community structure. And so even though I presented a lot of stuff here, it's not that exciting yet until we start getting some post-dam removal data. And I'm hoping, well, the data will be what it will be. So I'm excited for whatever it ends up being.
It sounds like it's a very, it's able to heal itself, shall we say, rather quickly after all this massive intervention, human intervention, dams and things.
Yeah, they're pretty resilient. And long as we're still not doing some other kind of major messing around with. Someone else have, yeah.
So after removing the dam 5,
Mhm.
do you expect to see the changes in the Nolan River, or do you think they would have to change that discharge from the lake?
I think there'd be minor changes, but I'm under the impression after seeing them discharge, that it's really scouring that system out. So some species are going to be able to hang on. I don't think there's going to be large abundances going to just get established. Where in the Green River, where the things are more stable, you can really start developing and getting some good numbers. I think you're always going to end up with kind of a transient type of community going through the Nolan. And when it comes to mussels, we did, I'll forget off the top of my head, 9 surveys in the Nolan, and only found 3 live mussels out of all those 9 surveys. And so it's just pure happenstance. Those mussels, I don't think stand a chance. Some of the fishes might be able to adapt, but I'm not optimistic about the Nolan.
Are you going to survey it after?
Yeah, I plan to continue the survey. I think it's going to highlight, the discharge practice that goes on by the Corps. Now they have to do some of it due to it's a large reservoir, fills up, but maybe better management might be a conversation along that line might be needed.
I think it's about
Did you look at any invasive exotics and or how would you expect dam removal to affect those ponds?
So there are big head carp, silver carp, which are non-native, and they are above Lucky Dam 5, but only, I think we got 2 individuals during all the fish work that we've done. I've done tons of fish work, and we only got 2 individuals. So I'm curious to see that the dam probably was a barrier to keep them from moving up, but they can get above, but the numbers were low. But now with the dam, that barrier being gone, I'm curious to see if they're going to become more proliferant within this impounded section or formerly impounded section of the Green River. That is the only exotic that we really encountered. Everything else, we're over close to 75 species of fish that we've gotten out of the green, and those carps, as well as the common carp, are the only exotics. Now, there's probably some natives that were maybe not necessarily native to that section of the green, but it's pretty minor as far as our abundances go.
If there are any goals about trying to bring back genetic diversity as opposed to just species richness or diversity in general?
I'm working with the professor at Austin Peay State University, and I was talking to her about what's going on here. And I said, don't you think it'd be a good idea to look at the DNA above and below the dam and stuff? So she is working on a proposal to address gene flow, I think is the thought. And the idea that we have is to match up the host fish for select mussels and look at how quickly, what are the differences above and below for these species prior to the dam, and then how quickly, what type of gene flow do we have, and maybe does that translate into better dispersal than for one of the select mussel species. The overall genetic diversity within whatever species, we aren't looking at that per se. Although we're getting a data set that we could do that for some of the select species.
We have a Zoom question as well. So, that's in the form of E.A. Sanchez. But asks, was there ever any kind of survey or concern when the dams were initially installed?
Nothing. Back in the 1900s, I would say no.
Any other questions?
Has anyone looked into how all of this is impacted reptiles and amphibians, or has most of the focus been on aquatic species?
It's just been the aquatics, and I being on the river as much as I have and just seeing all the turtles, I thought it'd be really cool if someone did some type of reptile type study there, but that's for them to do. I gotta draw my line somewhere.
I guess this is a question I'll follow a similar path, but as a follow-up, the surrounding like area shrinking, at pre-removal post the ecology change like in any significant way besides the lake stream thing falling down.
I have not detected that at all. I think on a very localized aspect, I'm sure it does. But at the scale and the way I have things set up, I don't think I'm able to detect that type of localization, that type of impact at that scale.
Any other questions? Okey-dokey, I think that's it. Thank you again, Mike.
Thank you.